All right, let's continue from where we left. Uh, you've seen there is this structure of this uh, uh, what you call like the amino acid residue, because it's made up of what was initially let me see. Okay, let me put it in red. This is the first amino acid, this is the second amino acid, and then this is the third amino acid. Yeah, so this was, they are joined together by the loss of water here, and then it's the loss of water here. Yeah, you can see there. So but just trying to draw those NH carbon with the hydrogen and those they're a bit cumbersome in trying to represent amino acids, especially when you have many of them on the same page. So scientists have been saying that it's much better to use the bond line structure than the normal common Lewis acid structure. So this structure which I've shown here is just being shown in a compact manner in this bond line structure. You can see here your NH2 is being shown here as NH2 here. Here there is the carbon, there is no other R group, so it's just being shown here as this carbon. You know, this is the carbon with two hydrogens. You understand, like this one here. So the carbonyl group is being shown here, carbonyl group down. This is the NH. This is the carbon with a methyl group. This is the carbonyl group. Then there is the NH. Then there is the carbon. This hydrogen is implied, and then there is the SH. And then the last one is the carboxylic acid. Can you see here? And this is the illustration of the fact that the R group here is hydrogen, so that is glycine. The R group here is C3, so that is alanine. The R group here is SH, so that is serine here. So this is an illustration of glycine joined to alanine joined to serine here. I've just tried to draw another one here. So there is the NH, there is the SH here. So this is serine. The next one is C3, that is alanine. The third one is hydrogen, so that is glycine. So this is the same as serine joined to alanine joined to, sorry, the third one is supposed to be glycine, yeah, because the R group is hydrogen here. Yeah. So this is the same as showing what? This is the same as showing NH2, SH, carbonyl group, NH, carbon with CH3, carbonyl group, NH, carbon, hydrogen is implied, so the R group is hydrogen, and then there's the carbonyl, and then there's the OH, yeah? You see, this notation is much easier to show, much easier to show than this complicated one, yeah? Especially when you've got many amino acids to show, so you're going to use this notation here, yeah? So with that one, you're going to look at the presentation of the various amino acids as they're being shown in your notes, yeah? Yeah, there are a couple of slides that are dedicated in your notes regarding the presentations of amino acids. So you're going to be looking at that one in your notes. So the next part of the course, of this lecture on the amino acids, I need to mention that proteins have got there is the primary, there is the secondary, there is the primary structure, which is the fundamental structure, just like what? Just like you going to school, yeah. There is, but now there is the pre-primary, yeah. So there is the primary, secondary, and then there is the tertiary, yeah. And now we are hearing there is the quaternary, yeah, beyond the tertiary. But as far as the education is concerned, everything else regarding advanced level is still in the tertiary level of learning. So regarding the regarding the proteins, the fundamental structure of how amino acids are joined, that one is called the primary structure. It deals with how one amino acid is joined to the next one. That's via a covalent bond called the peptide bond. That is called the primary structure of the protein. Now, you're going to see that there are other intermolecular forces that are involved, which I've shown here. Like I've shown a structure, one strand of an amino acid here, and I've shown another here. I'm going to see that when you're going to have an electronegative element like oxygen, yeah, it is next to a hydrogen which is joined to an electronegative element, in this case like nitrogen. Then there is a hydrogen bond that is formed here. In this case, there is the nitrogen which is electronegative. It contains hydrogen, and then that hydrogen is next to an electronegative element which is oxygen here. So there will be what? Hydrogen bond here. And so the story here continues. So between one chain of amino acids or the peptide, and then if you're going to have another one close by here, then there are going to be many hydrogen bonds that are going to be formed. So the joining of the structure to which is held compact by the hydrogen bonding, and in some other cases you can have a case whereby there's the sulfur plus another sulfur, they are joined together with the loss of hydrogen to form a sulfur-sulfur bond and a disulfide bond. That is the secondary structure of protein, in which the peptide bond and another peptide bond chain, they are held together by intermolecular forces. That will lead to the secondary structure of the protein. Then, as I mentioned, you can join one amino acid to the next through something called a peptide bond. Up to a certain level of the peptide bonds, that will still be called a peptide. A peptide has got a smaller number of peptide bonds. But if you're going to increase the number of peptide bonds, then it will be called a protein. So a peptide is essentially a small protein, yeah, and a peptide is made up of amino acids. So I want you to see the structure here. I mean the relationship here. So the overall chain of the amino acid that is joined, that contains intermolecular forces plus the primary bonds of the peptide bonds. The overall structure, it's so complex to really show. It gives the tertiary structure of the protein. So the tertiary structure of the protein is the complete structure, and what scientists have done, they've tried to use the visualization programs to which you're going to see in your notes. If you're going to see from the tertiary structure of the proteins, they may look slightly a lot of complicated structures, but it may look like a ribbon, all those kind of things and the rest, but that is the three-dimensional structure of the protein. And the way it looks matters, because that's how it's going to accomplish its biological function. For example, if it's going to be acting like a catalyst, like in this case, a biological catalyst is called an enzyme, yeah? If you're going to catalyze a reaction, then the molecule to be catalyzed for the reaction to go slightly faster or much faster has to fit into a certain depression within that three-dimensional structure called the receptor pocket. And once it fits exactly in that pocket, then the reaction, then the biological process or the catalysis is going to take place. That's when the enzyme is going to work. Yeah. So what else? I've introduced what the enzymes are a protein catalyst. Yeah. Catalyst proteins that act as biological catalysts. So that is the three-dimensional structure of the whole protein, is called the tertiary structure of the protein. Yeah. So in other words, is that for most of the proteins, the tertiary is the final complicated structure. Primary is made up of the amino acid linkages. Secondary is when they are linked up with the intermolecular forces. And then the tertiary is the complete structure of the protein. But there's some selective, I'm talking about selective type of proteins. I think for this one, I'm just going to delete this part here. For a selective type of protein, you can have, if I put that circle representing what? Representing a tertiary structure of the protein, the complete structure of the protein, you can have probably, that's a second tertiary structure of the protein, probably that one representing a third one, and probably a fourth one. So in this case, you've got 1, 2, 3, 4, 4 units of the tertiary structure of the proteins, yeah? And then these ones, they are linked together by a metal ion at the center. Maybe I put M plus, yeah? So they are joined together through intermolecular forces. So this metal ion, metal cation, it links together a number of tertiary components, tertiary units of the protein to make up something called the quaternary structure. And just to give you an overview, they are just as selective, for example, like hemoglobin, yeah? Hemoglobin is made up of the central ion, cation, and it joins a number of tertiary structures of the protein to make the quaternary structure, which I'm going to be calling hemoglobin. Hemoglobin has got in this case, the metal ion is called is ion, and it is joined to a number of tertiary structures. The other one, which I think I'll just mention, or which I think you've heard of, is this one, which I'm going to call chlorophyll. Chlorophyll has got the metal magnesium. Yeah, has got, I think the metal is magnesium. It's joined to tertiary components, so they form something called the quaternary structure. But so the quaternary structure is not there in many proteins. It's only a selective type of proteins which can only do their biological work when they are joined together by a central metal ion. Yeah, but in general, tertiary structure is normally the final structure of the protein which does the biological activity. But in some selective proteins, they go to the quaternary structure. So you read the notes about the uses and the various proteins like fibrous, globular, membrane proteins, and the rest, and enzymes and the rest. Yeah. So you look at those notes, part of the notes, and I think you'll be able to understand that. So I hope you've really understood about this section on the protein. So these 3 video series will just help you understand what is contained in the notes regarding the lecture on the protein. So in a short while, I'll just mention about carbohydrates, and the carbohydrates is usually a very complicated structure, but I'll just mention a few aspects so that for those who are doing biosystems, they'll get time to look at the more complex part. But for our case, because the course on the carbohydrates is quite wide, we'll just mention a few things, and then you look at the rest of the notes regarding the carbohydrates. But for now, I think that's the much. I'll talk about the proteins, and in a short while, I'll give an overview on carbohydrates. So thank you very much. I hope you've seen this course enjoyable. Okay, all right.